Dear students, welcome to our Chocolates class. This is an exciting journey into the chemistry of life. Let's begin. Have you ever wondered what you're made of beyond just cells and tissues? The answer lies in biomolecules, the fundamental building blocks of life. Carbohydrates act as quick energy providers, fueling every thought and movement. Amino acids combine to form proteins, which build and repair our bodies, shape muscles, and function as messengers. Lipids serve as long-term energy reserves, form protective cell membranes, and provide insulation. Enzymes, meanwhile, are the catalysts that make life's reactions possible, speeding up processes essential for survival. Together, these molecules form the very foundation of our existence. So here's a challenge. Proteins, carbohydrates, lipids, and nucleic acids. Can anyone guess what each of these structures might do in the body? Let's dive into this fascinating exploration of the molecules that make us who we are. And remember, biomolecules, the chemistry of life. What are biomolecules? The term biomolecule is derived from two words. Bio, meaning life, and molecule, meaning a chemical structure. Therefore, biomolecules are the chemical compounds that occur in living organisms. These are not rare or unusual substances. Rather, they are the fundamental components that enable life to exist and function. Biomolecules are found in every living organism, including plants, animals, fungi, and microorganisms. Biomolecules serve several vital roles. They form the structural framework of cells, cell membranes, and genetic material. They also participate in essential life processes such as growth, metabolism, repair, and reproduction. In fact, without biomolecules, life would not be possible. Why are biomolecules essential? Every living organism depends on biomolecules for survival and proper functioning. For example, a growing child requires amino acids to build and repair muscles and tissues. A runner depends on glucose as a source of energy during physical activity. Every new cell needs DNA to store and transmit genetic information during cell division. Thus, biomolecules are the basic building blocks and functional molecules that sustain life. Living organisms are not made of special or unique elements. They are composed of the same elements found throughout nature. The magic of life lies not in the elements themselves, but in how they are arranged and combined. The four most abundant elements in living matter are carbon, C, hydrogen, H, oxygen, O, and nitrogen, N. A useful mnemonic is CHON. Importantly, carbon and hydrogen are found in far higher relative abundance in living organisms than in the non-living environment, which reflects the organic, carbon-based chemistry of life. Biomolecules are organic compounds synthesized by living cells. Living cells actively produce these molecules through metabolic processes. This distinguishes biomolecules from inorganic minerals or simple compounds. Why do you think carbon is so central to life? Analysis of living matter. To understand what living organisms are made of, scientists must analyze them chemically. The two major approaches used for analyze chemically are 1. Chemical analysis and 2. Ash analysis. Chemical analysis refers to methods that identify and quantify both organic and inorganic compounds in living tissue, including techniques like chromatography and spectroscopy. Ash analysis, on the other hand, specifically helps identify the inorganic mineral content by burning the sample and examining what remains. These two approaches are complementary. Together, they give a complete picture of a living organism's composition. Ash analysis is a classical technique used to estimate the inorganic compounds present in living organisms. First, the sample plant or animal tissue is prepared by grinding and drying. Next, it is placed in a muffle furnace and subjected to ignition or combustion at temperatures between 500 and 600 degrees C. All the organic material, fats, proteins, carbohydrates, is completely burned away. What remains is the ash, which contains inorganic minerals like calcium, phosphorus, potassium, and magnesium. The ash is then collected and subjected first to qualitative analysis, which elements are present, and then to quantitative analysis. It means how much of each element is present. Organic compounds contain carbon and hydrogen, which escape as CO2 and water vapor when burned. The mineral salts do not combust, They remain as oxides or carbonates in the ash. Elemental composition, Earth and human body. In the Earth's crust, oxygen is the most abundant element, 46.6%, followed by silicon 27.7%, calcium 3.6%, sodium 2.8%, and magnesium 2.1%. Hydrogen is present in low amounts, 0.14%, while carbon and sulfur are negligible, 0.03% each, and nitrogen is almost absent. In contrast, the human body shows a very different composition. Oxygen is again the highest, 65%, followed by carbon 18.5%, nitrogen 3.3%, and calcium 1.5%. Hydrogen is present in moderate proportion, 0.5%, sulfur in low proportion, 0.3%, sodium 0.2%, and magnesium 0.1%. Silicon, though abundant in the Earth's crust, is negligible in the human body. This comparison highlights that while the Earth's crust is dominated by inorganic elements like silicon and calcium, the human body is rich in organic elements such as carbon, oxygen, and nitrogen, which are essential for biomolecules and life processes. Trace elements and their functions. Even though certain elements are present in only tiny amounts, called trace elements, they play indispensable roles in cellular function. Let's study certain specific trace metals in the biological systems they support, which is a critical concept in biochemistry. Manganese is found in the mitochondria, where it plays a role in antioxidant defense. Molybdenum is a cofactor for the enzyme nitrogenase, which is responsible for nitrogen fixation, the process by which atmospheric nitrogen is converted into ammonia for use by living organisms. Copper is part of cytochrome oxidase, an essential enzyme in the electron transport chain during aerobic respiration. Magnesium activates many enzymes and crucially is the central atom in chlorophyll, making it essential for photosynthesis. Iron, Fe2+, and copper, Cu2+, are found in cytochromes, proteins involved in electron transfer during cellular respiration. Finally, the order of cation concentration inside cells. K+ greater than Na+ greater than Ca2+. Chemical analysis using trichloroacetic acid, TCA. While ash analysis reveals inorganic content, chemical analysis using trichloroacetic acid, TCA, is used to identify and estimate the organic compounds within living tissue. This is a crucial technique for studying biomolecules at the biochemical level. First, the organic sample is crushed and dissolved. Then TCA is added. TCA is a strong acid that denatures and precipitates proteins, separating them from smaller molecules. The mixture is heated to break down components further. Centrifugation separates solids, macromolecules, from liquids, micromolecules in solution. Filtration then removes the solid residue. Finally, the filtrate is analyzed using techniques such as spectroscopy or chromatography. T-trichloroacetic acid has 3 chlorine atoms attached to an acetate group, making it a powerful denaturing agent. Analysis of chemical composition. This part summarizes the outcome of TCA treatment and introduces 2 critically important fractions that biologists work with when analyzing cell contents. When living tissue is ground in trichloroacetic acid, the resulting slurry is filtered to yield 2 fractions. The acid-soluble fraction is the filtrate, the liquid that passes through the filter. It contains the smaller molecules or micromolecules which are not precipitated by TCA. Examples include amino acids, glucose, and nucleotides. The acid-insoluble fraction is the residue left on the filter. It contains the macromolecules, proteins, polysaccharides, nucleic acids, and lipids associated with membranes. Chemical composition of living organisms. Living tissues contain and organized into 3 broad categories: organic compounds, inorganic compounds, and water. Organic compounds are the carbon-based molecules produced by and within living cells. Carbohydrates, proteins, lipids, and nucleic acids. These are the biomolecules that we have to discuss in depth throughout this chapter. Inorganic compounds include water in its ionic context, minerals such as calcium and phosphorus, and various ions like sodium, potassium, and chloride. Though not organic, these are equally vital to life. They regulate pH, enable enzyme activity, and carry electrical signals in nerves and muscles. Perhaps the most important point is that water is the most abundant chemical in cells. On average, water makes up 70–80% of a cell's mass. It is the solvent in which all biochemical reactions occur. Without water, life as we know it could not exist. Acid-soluble and acid-insoluble pools. This part consolidates the TCA filtration process into a clear table format and introduces key terminological distinctions that we must know. After TCA treatment and filtration, the sample divides into two pools. The acid-soluble pool, the filtrate, contains micromolecules, amino acids, sugars, nucleotides, whose molecular mass does not exceed 800 daltons. These are also called biomicromolecules. The acid-insoluble pool, the residue, contains macromolecules, proteins, nucleic acids, polysaccharides, with molecular masses greater than 800 daltons. These are described as biomacromolecules. In case of lipids, they are present in the acid-insoluble pool because they are associated with membranes, but they are not strictly macromolecules in the polymeric sense. Their individual molecular weights are not as high as proteins or nucleic acids. Primary and secondary metabolites. Primary metabolites are compounds that are directly involved in the essential metabolic processes of life. They are required for growth, development, and reproduction. Examples include amino acids, building blocks of proteins, sugars, energy sources, chlorophylls for photosynthesis, and heme, the oxygen-carrying component of hemoglobin. Every living cell produces primary metabolites. They are universal. Secondary metabolites, in contrast, do not have a direct role in primary metabolism. They are not essential for the organism's immediate survival, but they often serve important ecological functions, attracting pollinators, repelling herbivores, or defending against pathogens. Examples include rubber from latex, drugs like morphine, spices, pigments, and fragrances. Particularly noteworthy is the mention of cyanotoxins, which can be used as herbicides and insecticides. Examples of secondary metabolites. Pigments such as carotenoids, the orange in carrots and the red in tomatoes, and anthocyanins, the purple in blueberries, protect plants from UV radiation and attract animals for pollination or seed dispersal. Terpenoids, including monoterpenes and diterpenes, are found in essential oils and resins. They give plants their distinctive scents. Alkaloids like morphine and codeine, derived from the opium poppy, are powerful pain-relieving drugs used in medicine. Essential oils such as lemongrass oil are used in aromatherapy and food flavoring. Toxins like abrin from rosary peas and ricin from castor beans are among the most deadly natural poisons known. Lectins such as concanevalin A are proteins that bind carbohydrates and are used extensively in research and immunology. Drugs like vinblastine from the periwinkle plant are used in cancer chemotherapy, and curcumin from turmeric is under investigation for its anti-inflammatory properties. Finally, polymeric substances like rubber, gums, and cellulose have enormous industrial applications. For academic convenience, the topic of biomolecules has been divided into clear, easy-to-follow parts. Part 1, Introduction to Biomolecules. Part 2, Carbohydrates. Part 3, Proteins. Part 4, Lipids. Part 5, Nucleic acids. Part 6, Enzymes. This structured division will help you study step by step, connect concepts smoothly, and revise efficiently. Remember, biomolecules are the chemistry of life. Master them, and you master the foundation of biology.
For tailored topics and curated content, connect with us today. WhatsApp 8639823875.